I will introduce our last speaker of the day, who is Dr. Xin Shu. Dr. Shu is a senior scientist and the director of pharmacokinetics in the therapeutics development branch of the Division of Preclinical Innovation here at NCATS. And she will be talking to us today about in vitro assessments of ADME properties of lead compounds. Welcome, Dr. Shu.
Thanks, Shireen. Can you hear me well?
Yes.
Good afternoon, everyone. Thanks for staying with me for the last leg of today's presentation. I've been in this kind of situation for several assay guidance planning workshops, so I'll be the last speaker. I have to cheer myself up by thinking, imagine you're really race. The last leg actually the strongest one, right? So thanks for stay with me for the last, the talk. We'll finish today's program together. The topic I'm going to cover is the in vitro assessment of ADME property lead compound. I will start with acknowledgement. As you know, our field is quite an established field. There's so many people working in this field very hard for decades. So the data we present here is a collective contribution for so many scientists, including my current colleagues and also the former colleagues. And also I'd like to thank Dr. Chung and Dr. Smith from San Bernard Prisby Institute for their contribution on the PharmacoConnect chapter in the assay guidance mining. So for my talk, I will start with the introduction, talking about the importance of the ADME PK concept in drug discovery and development. Then I will really talk about the rationale, how we design in vitro assays that be used to address the key ADME issues during discovery or late-stage development. We'll go over some common assay briefly, and we'll end the talk with some take-home messages. As we all know, from a great idea to the final product will take a long process with so many hurdles there. So the ADME is the one we're dealing with today in this talk. So before we talk about the ADME assay, probably I should talk about what is pharmacokinetics. When I was in the graduate school, I thought of pharmacokinetics, you write a math equation, we derive so many differential equations and build models. That is, I thought was pharmacokinetics. But then I joined the company in the pharma, then you see people interpret it very differently. You have a biologist come to you with a tube in his hand and say, can you measure pharmacokinetics for me? I look at the head and I said, what tube in pharmacokinetics? It just really shocked me in a way, say how people view what is pharmacokinetics. So now over the years, I gradually realized, pharmacokinetics actually is a really broad term. It just many meaning there. People not in the field, probably they could interpret from whatever needs they have. But for us, pharmacokinetics is a science that describes the movement molecule in the body. How do we monitor this molecule then? We collect plasma samples over time, measuring the drug concentrations. Then based on this, what we call the concentration time curve, we'll derive the mathematical equations and describe this curve. And then use these mathematical equations, we come out with some parameters. They really describe the disposition kinetics for this molecule. Why pharmacokinetics is important? This show in the early 90s, in the clinical trial, the failure due to pharmacokinetics almost like 40%. Realize it is a major issue for the drug fail in the clinical trial. All the pharmaceutical companies start move pharmacokinetics early on, trying to minimize this risk. So you can see after decade effort, Now into the clinical trial, the failing due to the pharmacokinetics is really reduced dramatically. This is another chart to show in the 208 and 209, the phase 2 successful rate is only 18%. The major failure is due to the lack of efficacy. But look at what is the percentage of pharmacokinetics failure is less than 1%. So really, we're very successful to accomplish select the good molecule, go to the clinical trial. So now back to this PK concept and ADME assays. How we do it? You give this plasma concentration profile. If this concentration too low, you're probably not going to see the efficacy. But if it's too high, you'll probably hit toxic region. So you want to choose the compound. They can deliver the profile, it's adequate, give you the efficacy, but not toxic and give you the side effect, you don't like it. So this curve is really controlled by so many detailed process, but we can briefly divide into the 4 process. It is absorption, distribution, metabolism, and excretion. By knowing all these the process classified, and then we will design some in vitro assays to address the key issues that govern all these process. Hopefully, based on the in vitro assay, we can select winning molecule. We call that is the drug-like molecule, and then we can spend more resource development as a later stage development. So that definitely will save the cost, and also really we have a better molecule sent to the clinical trial. After we get all these data from the in vitro assay, then we can really come back to do this, the modeling is called pharmacology based modeling. Then we can predict in vivo pharmacokinetic profile based on the in vitro assay data. In this talk, we probably just focus on the small molecule. For the small molecule, the oral administration still is the most convenient route for patient. For drugs swallowed by patient by mouth, the drug molecule has to go through several barriers before they reach the systemic circulation. The first biological barrier probably is the stomach. We know the pH in stomach usually is pretty low, especially in the fast status, somewhere between 1 to 2. If your compound is not stable chemically, it's likely to be decomposed in the stomach, so they won't get the drug reach the circulation in the blood. And also, if the pH is low, for some acidic compound, they're probably going to precipitate out in the stomach. Then they will further reduce your absorption chance when they go through the intestine. That's a side of absorption. We call this, the biologic barrier 2, that is the intestinal membrane. For the intestinal membrane, although they have some the pores there for small molecule, they can go through. But these days, Medican, if you talk to Sam, these days Medican molecule made, it's really relative size-wise is bigger than the older days. You have a molecule weight less than 300. They probably can pass those membranes through some pores there. We call this a paracellular pore pathway. So most compound actually have get into absorb pathway. It has a membrane through what they call this a transcellular pathway diffusion. So in this way, then they have to really be a certain permeable. They can pass this membrane in the intestine. There's some other mechanism involved, such as the active transporter. We call that influx uptake transporters. For some big molecules, they even go through the other mechanism called endocytosis, pinocytosis. In the intestine, there's another barrier there we call barrier 3, that is the drug metabolism or efflux transporter. We talk about the influx transporter for uptake, but counterpart to that, actually, even the drug get into the intestinal membrane, but they can pump back from the through the efflux, such as from PGP transporters. And also some drug, when they move along the intestinal lumen, they're not stable. There are so many drug metabolic enzymes there in the intestine. They can decompose the drug, so we don't have the parent drug again reach the blood circulation in vivo. So the next organ the drug molecule encounter will be the liver after intestinal absorption. Most mesenteric vein from the intestine drain into the portal vein. So the drug is really carried in the portal vein, go to the liver along with these sinusoids. The liver actually is the major organ for drug metabolism. They're very rich, tons of drug metabolism enzymes there. In addition to the drug metabolism, there's another mechanism, there's the biliary excretion, so they can dump the molecule to the bile and excrete it back to the intestine and into the feces and excrete it out. So now you can see before the compound reach the blood circulation, they have to pass several barriers there: stomach, intestinal membrane, intestinal metabolism, efflux transporter, and the liver metabolism, biliary excretion. And overall bioavailability is a product of all these little, what do you call the available fraction drug pass through that barriers. So the bar is pretty high. Then we really need to see if we can design a molecule that can successfully pass all these barriers to ensure they can reach the systemic circulation. So next process is distribution. After blood in the blood circulation, they will really penetrate the tissue, especially target tissue, to be active. So if you look at key physical chemical properties that govern the distribution, probably you will count the size, charge, lipophilicity, and so on. Other factors we need to consider is like protein binding in the plasma or even the RBC partition. Think about these are probably as reservoir hold on the drug molecule. Only the free drug they can really exchange into the tissues and be uptake and distribute into tissues. So other factors we can look at is really in the binding on the tissues. Either it's a target, a specific binding or non-specific binding in the tissue. All these bindings they really govern what is the free drug concentration available. It's a free drug concentration usually will be really switched around, move around, and then either be recirculated back to the liver, be metabolized or excreted and so on. So these are the process we're looking at. Now the next process is metabolism, what we call biotransformation. You already hear from Sam's talk. There are so many metabolic pathways that really the important ones, the phase 1 reaction. There's the oxidation, reduction, and the hydroxylation. The purpose of these reactions really adding the polar functions to the molecule. So the increase the solubilities and this molecule can be excreted. The phase 2 reactions, conjugation reactions, further adding the polar cofactors there, like UDPGA for the gluconation, sulfate for a sulfation reaction, or glutathione conjugate. So those are the molecule be readily excreted. And sometimes all these phase 2 reaction also considered as a detoxification reaction because all these conjugate, usually the potency is less than the parent drug. Sam briefly mentioned about the P450. Yes, that is the most predominant enzyme for many the drug metabolism reactions. P450 really mediate so many reactions there, like aromatic hydroxylation, lactic hydroxylation, epoxidation, alcohol oxidation, etc., etc. So that's why we start pay attention to this enzyme when we design the in vitro assays. So once we start address the P450 mediated reactions, then we notice actually non-P450 mediated metabolism these days play a more important role now. You know, our body is pretty smart enough. Once you block something, they just force them to go to the other enzyme systems. So more and more we can see other enzyme system now we cannot ignore. We have to pay attention with screening. Why all this is important then? Because the different enzyme, they have different location in the cellular. You know, we talk about the liver, probably hepatocytes, but for the cells, the P450 is really mainly located in the microsome fraction. However, for other enzymes, for example, aldehyde oxidase, they located in the cytosol fraction at the soluble fraction of the subcellular locations. So knowing all these help us think about how we design the in vitro assays with what matrix. Now we dive in more deep in the P450 because it's such an important enzyme. There are so many isoforms in the P450s, such as 1A2, 2C9, and 2D6, 2E1, and 3A. And these enzymes widely distributed in the liver, in the body. Liver be the most important organ, but nevertheless, other organs such as kidney, lung, and intestine also have P450s. For example, in the lung, the 1A2 is the major enzyme. A lot of enzymes found in the liver actually also in the intestine. Although the Vmax or the total enzyme amount probably less. The intestinal metabolism is very become important when you deal with the low dose. But when you have high dose, probably either saturate intestinal metabolism than reach the liver. So again, the liver probably that's our main focus when we're dealing with the drug metabolism stability. P450 is known to be polymorphic, and also we know there's a species difference. So when you do the assay screening, you can look at what species you're dealing with. Ultimately, our goal is the human, but you probably start with some animal study first, and then you have to understand what is the enzyme activity, what's isoform in what species. This is the chart show the P450 isoform in human livers. Not surprisingly, 3A4 is the major one. It's the orange color here. And the 2C family actually also is quite a big one. It's the color here. The 2D6, this is the one, this is the purple one. It's not that, in terms of quantity, it's not that large. But however, if you look at this chart here, this shows the cytochrome P450 isoform involved in the drug metabolism in the approved drugs. You can see, not surprisingly, 3A4 is the major one, but 2D6 actually is quite important. The 2C family also is important, but again, in terms of distribution here, we know they're one of the major isoform responsible for drug metabolism anyway. So it's not totally surprise. So the last, the process we talk about is excretion, the ADME, the E. So really the two, the process we're looking at, one is the biliary excretion, one is the renal excretion. Although sometimes the lung from the inhalation also can play some role there, but not so many molecules be eliminated by that way. Still, it's through the biliary excretion or renal excretion. So for the biliary excretion, again, the factors we talk about, the size, lipophilicity, and the transporters. Really, the transporters these days, people pay more and more attention to understand the role, how drugs being pumped out through this excretion mechanism. After I describe all these ADME process, then you can see there are so many factors that govern all these process. Then we can design the in vitro assays to address all these process. Then that's how we rank, select the winner molecule. For example, for the absorption, we can have the assay to deal with the permeability. Now we can look at if it is the efflux transporter substrate, and also we can assess its solubility. For the metabolism, we spend quite time to talk about, P450s. Definitely, we have the assay designed to assess the, cytochrome P450 mediated metabolism. But we can also look at, other metabolic stabilities. We probably going to talk about, some plasma stability for prodrugs later on. Once you have so many assays there, running all the assay for every compound can be very costly. That's why we group our assay into the two category. One is called the tier 1 assay. It is a high throughput screening assay. You already hear enough today in the early discussion. The tier 1 as what we call a high throughput assay usually run at a very simple condition, probably just one concentration or one time points. But nevertheless, it is an automated assay with a rapid turnaround. You can handle a large compound very quickly. Then for project-specific needs, we have a tier 2 assay. So they really what we call a customer assay to address project-specific needs. These assays sometimes not a very high throughput fashion, but nevertheless, they will really provide useful information to guide Medkind synthesize a new compound. And we can have the really the select compound for the later stage drug development. Through this screen paradigm, I also want to add here is the initial in vivo PK study, probably in the rodents. We need to quickly establish what's the relationship between the in vitro assay and the in vivo, and to identify what are the key driver for the disposition kinetics for the scaffold selected. So once we know the key assays either is due to the metabolic stability or permeability, then we can heavily use the in vitro assay to drive the structure optimization process. For the tier 1 assay, as I mentioned, it's really the purpose for ranking compound. It's really quick assay. We usually including this assay in the tier 1 assay on the solubility, the permeability, and the liver microsome stability. Sometimes we do add inhibition studies there, as we know the 3,4 is the dominant isoform for drug metabolism to assess, drug-drug interaction potential in the clinical application. So we're probably trying to avoid the compound that has very a liability on the CYP inhibition. At NCATS, this is we use a TCAN automated platform for our high throughput screening tier 1 assay, and we have permeability assays called PAMPA. It's artificial membrane assay. This is the membrane loaded. You have a drug from the donor side. You stir it at room temperature for 30 minutes, and hopefully the drug molecule passes the membrane. Then you measure the drug on the receiver side. So it's really how you assess this permeability. We use this artificial membrane to mimic the cell permeability. We also determine what you call the kinetic solubility. That is, drugs dissolve in the DMSO and then you spike into the buffer at pH 7.4, and you stir at room temperature for 6 hours and go to the filter, then measure drug concentration. Another assay we talk about, metabolic stability. In the tier 1, it's maybe just one concentration. In our lab, we use 1 micromole. And it's a very short assay. somewhere between 15 to 60 minutes. In our protocol, we use 15 minutes for tier 1. Then for the CYP inhibition, probably we can just focus on the key isoforms like 3A4. At INCAS, we have this rapid fire assay developed for the 3A4. We know the assay performance is very important when you screen so many compounds. So in our tier 1 assay, we have every place have controls there. We monitor performance of control substrate in each place over time. So for the 4 assay we commonly run in the tier 1, that is a PEMPA at pH 7.4. Later on we add PEMPA at pH 5, and the kinetic solubility and the microzone stability assays. Over the years, these are the results. We have data for over 500 places there over the last about 7-8 years. And we look at what is the MSR for these assays. So you can see our MSR for these assays are not that bad. Usually we consider if MSR less than 3, we consider it good. So for our assay, somewhere between 1.5 and less than 3. Usually, say for example, if it is very low, the PEMPA parameter very low, so it's no we can calculate it. So you only calculate for moderate and the high controls there. So that's the value we closely monitor over time. This is the data we report back to the team, in vitro ADME properties, together with this compound list. Monthly, the team meeting, you probably show, what's the results you have, and then we use a color code. The green means good, the red color means not so good, and the somewhere in between is the yellow. So it's visually you can really pick up the one which direction to go in the structure optimization effort. For the tier 2 assay or customized assay, as we said, it's really address project specific needs. These are not really high throughput screen assay, sometimes a cell-based assay. For us, cell-based assay not come as so easy. We will need a lot of labor and resource to run these assays. So that's why we don't screen for every compound. Only the select compound will run the tier 2 assay. I will give some example to talk about the stability and also the might idea how we run these assays. The stability in the blood and plasma. So why we care about it? We talk about the liver is the major organ, but sometimes the clearance report back, your clearance so high, you see the hepatic blood flow rate, you know something else there. So that's where you look at, what else could happen there for extrahepatic biotransformation. One of the possibilities could be the stability in the blood and plasma, especially for the prodrugs like esters and so on. If your molecule had a function group like AMAs, lactone, and you probably should really look at the stability in the blood, in addition to the liver. Recently, I got a request from the project team. We tell them they made a prodrug for the antiviral project. They said, the microzone stability or cytostability is very good, but they still want us to run this one, the blood stability. We said, yes, we needed to look at, the stability in the blood and the plasma. It's really for the extrahepatic metabolism. But however, if your compound is really stable in the liver, the likelihood just purely metabolized by the plasma enzyme probably is not that high either. So they didn't really believe that. So we did the plasma assay too, and turned also it's pretty stable for their compound. Another case I can mention about it, we all know the remdesivir during this COVID-19 pandemic crisis now. So if you look at the enzyme metabolism for the remdesivir, this is a prodrug, right? The carboxylesterase is really the enzyme contribute the conversion of remdesivir prodrug. 80% of the biotransformation is mediated by the carboxylesterase. But actually, the carboxylesterase 1, that's the enzyme reported in FDA insert there. This enzyme actually is located in the liver. So that's why I was trying to say, yes, we will look at plasma stabilities, but nevertheless, we can still use the liver enzyme metabolic stability guiders too. So the next example I'm talking about is the MedID to get used for the data medical people like us to provide. They want to know what are the soft spot or vulnerable spot for the drug molecule being metabolized. The example I present here is called for the FOP project. The FOP is a rare disease, ultra rare disease, affect the patient 1 in 2 million. So when our collaborator from Mass General Hospital present the lead compound, that's LDN-193-189 to us, this is structure presented here. We know this molecule will have some liability based on our past experience. So the first experiment we conduct for this one is the MedID study. So indeed, when we get results back, we do see the several liability for this molecule. One is they form the aniline. The aniline is known associated with the safety concerns. Another one is we use this what we call potassium cyanide as trapping agent to trap the intermediate reactive metabolites. Indeed, we observed 2 cyanide adducts for this molecule, suggesting they form the intermediate reactive metabolite. Another thing we noticed is this molecule formed this hydroxy metabolite is through this AO-mediated metabolism. We already talked about the AO these days more and more play the role in the drug metabolism there. The AO, why we care about it? AO is species dependent. For example, a dog doesn't have the activity. The dog is known as a non-rodent species for tox study. So if the compound have a major pathway mediated by AO, the dog will not cover the toxicity for that metabolite. So that really added the extra risk when you choose the dog as a non-rodent species. Then you probably say, okay, how about I choose the monkey? Right? That's another common non-rodent species for the tox assessment. In the monkey, actually the AO activities is very high. So you go the other extreme, then your parent drug concentration will become pretty low. Then you worry about, the parent drug concentration, will that cover the human exposure for parent drug? So that's why, it is a pretty challenge if the AO is a dominant pathway for your molecule. These days, I do see people trying to push for mini-PEG, but the mini-PEG, the size actually is not that small. So if you use a mini-PEG for tox study, the quantity of material required will be pretty high. Did I stuck here? Hello?
We can hear you, but maybe the slides are frozen.
Yeah, so what happened then?
Are you on VPN, Shin? Maybe you might want to disconnect from VPN.
Oh God, sorry. So how can I go back then? Okay. It's okay now? Can you see well?
Yes, it's
That looks
the
great.
next slide.
Okay, great. Sorry about it. The next assay I will briefly cover is the detailed CYP inhibition assays. We talk about the high-throughput screening for the CYP inhibition, but for the lead, if you really choose the lead going to the clinical trial, you probably need to assess the detailed CYP inhibition, because these days we don't just give drug one single drug. You probably give several drugs together. So DDI assessment become very important. Those kind of studies pretty expensive. And so you want to know what is really the details, the inhibition, your new drug to potentially impact on the co-administered drug. So you probably have to look at all the isoforms recommended by what we call FDA panel, including all the isoforms, the major isoforms there. And in the assay, you probably have to do the detailed concentration range to cover what you project the human concentration range there, not just a single concentration anymore. In addition to the competitive inhibition, you probably also to look at the irreversible inhibition, or we call the time-dependent inhibition, and determine what is the Ki for the inhibition reactions. The last, second last assay I'll talk about is CYP induction. The CYP induction needed to use the cells. That's why it's not really high support for us here. You really need to very careful when you select the compound when you do the induction. You probably also should know what is relevant concentration you look at when you select the concentration to the induction. Sometimes people go outside with the CRO ask to do the induction studies there. So when you go through that route, you probably should really talk to the CRO about your project to really understand if you choose the right concentration, because you don't want to cause false alert. That's really damage your project, or you don't want to omit it. So this is very tricky how much you run, and especially it is not the cheap experiment. For induction assay, there are two ends to read out on the assay. One is the mRNA assay, really to measure the mRNA by the qPCR. Another, it is really measures through the probe activity, use the mass spec. So either way, especially probably the combination both will really assure this is a true phenomenon. For the details, FDA actually doing a very good job. They posted this online guidance. If you're really in the drug development stage, you probably should really reference those FDA guidance. This is the last assay I'm going to talk about, is transporter. Transporter, it's really the air continue evolving and add more and more transporters there. People start really pay attention on transporters. It really impact the tissue distribution, absorption, and also the elimination. So transporters, again, it's a cell-based assay. It's not easy assay for isotope screening. You probably need to focus on the important one. For example, for absorption, you probably look at intestine. What is the transporters in the intestine? They really impact your absorption. Some are efflux, some the influx. And for the liver, again, you look at what is for the uptake, and also what is really for the excretion out of the bile. Similar for the kidney, what is really the uptake, release absorption process, and versus really the pump out. The FDA have this guidance on the transporter evaluations. The guidance came out in 2012. In that one, they have this the flow chart guide you when do you need to look at transporters and what transporters you should focus on. I thought this is very practical. For example, if you measure the bile, look at what's the percentage parent drug excreted into the bile. If it's greater than 25%, probably you trigger it to look at the transporter involved. Similarly for the renal excretion, these days when I do the IV dosing, especially in large animal, I do collect urine samples here, trying to get some sense on the renal excretion of parent drug. If you look at the renal excretion of parent drug greater than 25%, probably that will trigger you to measure more detail on the transporters. Later on, FDA have the update guidance on the DDI assessment, including the transporters. In this version, they divide it into the two guidance. One is for in vitro metabolism transporter to assess DDI. The second one actually is for human clinical DDI assessment. So there are lots of information there. If you're interested, you probably can read more in detail there. So why so challenging for the transwell assays? Because transporters, many transporters unit directions. They have to really to see the directions. So use the transwells there. Then you have the assay. You have the spike drug from both sides. One A to B and the B to A and so on. So then all these cells take time to really qualify for the assays. And the current, most the wells we have probably either 24 well plates. Now more and more we start adopting the 96 well plates for transwells. So for transwell assays, you have need to meet so many criteria and controls. You have to have positive for the efflux controls, and you have to have high permeable markers, and also the low permeable markers, and the tiers measurement. And when we start work in the transporters, currently in our lab, we really just work on the CACOs. We realize for the tiers, automated instrument for tier measurement is not there yet. So we work with the applied biophysics. They have the ultimate tier measurement for 24 wells. So we work with them now to develop the device. They can measure the tier for 96 well transfer cells. So the beta version of the instrument is available now. So for people interested, you may contact the company. Again, Applied Biophysics, if you want to try it. Once we have all the in vitro assay data, we really need to correlate with in vivos. We know there are differences between these two systems. One is isolate system, then is a complete system. One is a closed system, for example, test tube. The other is an open system. You have in, you have out, dynamic. Everything is a change with time. So the in vitro is a simplified system, and the in vivo is a more complicated system. You have other factors there, so you should really pay attention. But nevertheless, in vitro assay is still very important for us to guide us optimize structure and select leads. For the in vivo part, I'm not going to go through detail, but these are the key PK parameters we usually use. For exposure, it's a Cmax and the AUC. For absorption, we use the bioavailability, and distribution, probably the warm distribution in the steady state. And for elimination, it's really the two processes there. One is metabolism and one is excretion. We use the clearance and the half-life. You can calculate by yourself with your own algorithm, but really there's a commercial software there you can use for PK parameter calculation. So the last topic I want to cover is in silicon ADME. I can see this before we say the future, but now I can see more and more companies actually start applied in silicon ADME. So it's really computer-based design prediction tools. There are some commercial software where they're available, but sometimes the fee is very expensive, or some software probably built based on limited compound numbers here. So that's why, when you try the software, still you have to really understand how that software is built. In our lab, as we said, over the years, we start build our own data sets. Then we start build our own model for each parameters. For example, we have this kinetic solubility model just published last year. This is based on over 10,000 compounds we collect from over 200 NCATS projects in 8 years. We developed this model, use this support vector classification model. In this model, we use 80 compounds as a training sets and 20% compound as testing sets. If you look at accuracy, we use this AUC of receiver operating characteristic curve, or AUC. Then for this, the solubility model, we can reach accuracy over 90%. This you can view this model on the website. So, so this is the exercise, the fun part. We say we have model. How good is the model, right? If you cannot convince yourself, how can you convince other people to use your model? So, beginning of last year, we have 90 compound because every week we screen the compound, new compound, right? So we have a 90 compound new collected data in early 2019. So we tried the two scenarios. The first scenario is that we adding this 900 compound with the existing model, and again divided 80% training set and 20% testing set. And then you look at the AUC, we still can achieve accuracy over 90%, just like the global model we achieved before. It's pretty good. But then we say if the customer going to use the model, they're probably not going to have the capability to rebuild the model with the new data there, right? They're probably going to use existing model and predict what happened to this new 900 compound, right? So this is the second scenario. So we just use the existing model, predict the solubility for the 900 compound that is newly synthesized. So we come out with the accuracy, the drop from 90%, but still it's over 80%. So suggesting the compound structure in this 900 compound, probably there's some different from the previous data sets. So next step we did is we look at more detail this 900 compound. We select compounds, they have at least, we select project at least have 20 compounds for that project, and then look at the accuracy for the prediction. So you can see some project will get very good prediction. We got almost 100%. This one definitely over 90%, right? So these are pretty good. But for some project, you get very poor. Then these are the scaffold, definitely it's not covered there. So our next step is really trying to help the customer build their scaffold specific one as a training set. Hopefully, give you better prediction on the new compound to be designed or to be made. So, nevertheless, for this local model, we have the average from this 8 project is about 77%. After we build all these data sets and the models, these special data sets, how we deposit our data. Recently, we start to upload our data to the PubChem. There's 4 assays already loaded there, so you can really see some data there. I have to apologize. Not all our data can load it. The data, though we have it, we build a model, but since a lot are active project, we cannot release the structure immediately. But it is our commitment. We'll gradually release whenever it's cleared. So that is what we'll continue doing. Currently, we have 4 assay upload to the PubChem now. Then we're working on additional one, the solubility, for example, we just got published, so we're going to load that one. Another data set we have is this collaboration with IQ Consortium. This one we can disclose all 5,000 compound used because all these 5,000 compound are most are from NPC library or some the one commercially purchased compound. So there's no patent sensitivity there. So we can load those data sets there for public use. So I will pretty much summarize my talk here is the take-home message. I hope through today's talk you believe in vitro ADM assay are important tools in drug discovery and development. They can guide the mechanism structure optimization, facilitate drug-like candidate selection, predict in vivo PK and DDI potential assessment. The high throughput assay is commonly used to support the discovery activity. The detailed assay is required to address project-specific the issues, especially for the lead candidate. We have to go to detailed assay. To make sure the assay data is good, have high quality, we need to have controls and replicate in the assays. So I'd like to thank you for your attention and stay the last minute with me. Any question?
Hi, Xin, thank you for a very nice presentation. Yes, actually, you do have many questions here. So with the few minutes we've left, I'm gonna go ahead and address those questions to you. So the first question is, do you think, so questions keep coming, so all right. Do you think that utilizing Keiko-2, MDCK, and PAMPA assays can help us better understand how to predict toxicological issues?
This is a very complicated question because you asked for toxic. If it's pharmacokinetics, I have more confidence. Toxic part is more complicated, right? And I probably let Mengheng to answer that question. For this, the Keiko, PAMPA, it's really the permeability assessment. Really to help us understand, will they be able to pass intestinal membrane? Or will they be able to pass the target tissue you're interested in? We did try our assay to mimic immune case. For example, for the Keiko, we're trying to have a different pH, like pH 5 mimic like intestinal pH, right? So that study is ongoing. Actually, we're working on the manuscript. Again, there are some category compound probably easier to have the prediction, but some are more complicated because you have drug metabolism there too in the Keiko cells too. So you need to really look at your metabolism. Then if you screen out the metabolism part, then just use the remaining compound, their stable compound, that Keiko permeability, and we do trying to correlate the PAMPA permeability with the Keiko, the stable compound correlability. We do get quite good correlation there for the permeability assessment. But for the tox, I'm sorry, I'm not so sure exactly what toxicity you're talking about, so won't be able to give a very general answer.
Thank you, Xin. We have a question from Yameng, who's on our panels list. So Yameng, I'm going to ask you a question, and if you'd like to comment more on it, you can unmute yourself. So let's see, I lost that question. Okay. He says, thanks for this great talk. Can you comment on whether covalent protein binders may have different PK properties? Oh,
yes, absolutely, yes. If you have a covalent binders, right, you see the field evolving. When I was in the pharma early, like in the 90s, that time, if you are covalent binders and totally you're out. We even screen out for the isogluconide. Isogluconide has a stability issue. They have covalent binding, so we're very cautious on this covalent binding because the covalent binding are related to the idiosyncratic reaction. That is the fear. But then later on, when we moved to Boston area in 2000, and I see people even use covalent binding as an approach in the therapeutic development. So if that's the case, So you have to tell the thing differently, because if your drug has colon binding, very likely to bind the plasma, right? How it assess colon binding? Actually, we use a plasma incubation. You already know how much loss, because plasma not so much drug metabolism there, right? Especially if you don't have those lipo function group molecule, then you know it's probably colon binding. Then what do you measure there then? You really measure its extractable concentration. You lost a lot there. So for those the project purposely use colon binding as approach, you probably should use toxicity, safety assay driven. Now they use PK driven, because the suspend PK is not going to help you much. If you're lucky enough, you have a radio labeled compound that can give you better in terms of quantitation. What's the molecule you're looking at, where they are. Otherwise, you just based on traditional organic extraction or assay mass spec for the pen drug, you probably miss them. That's my experience. But it would be nice to know early on what you're dealing with, your assay, and how to interpret all your ADME data sets. It will be very different.
Thank you, Shin. One more question, last question. There's actually more questions. Maybe I'll ask this question. If you have time later, you can check the chat, the Q&A box. So Sam is asking, what do you think about metabolism assays to produce and isolate metabolites and test them for activity in the primary assay?
That is a very good topic too. Science-driven assay development, right? We know the ID is very labor intensive, but these days we have a good software actually guide us very quickly. You probably not know a lot of detail, but at least you can capture some few soft spot very quickly. So we have a young scientist in the lab. They're very smart and have lots of image. So they said, they come to me, they said, we have this tier 1 assay for metabolic stability, right? If Metacam present this scaffold to you, they're not stable there. Say in vitro half-life less than 5 minutes. Could we automatically start MetID with our software? So we are really approaching that direction. Hopefully, if we have more
Matt, maybe you can engage the happy hour people as I'm finishing up in the main room.
Oh, I didn't even see that they.
Yeah, we have a couple connecting.
Abigail, we can hear you. We can hear you at the main session.
Hi, Matt. How you doing?
We can hear you guys at the main session. So you might wanna log out from the main session if you're in.
I muted them.
Okay, great. Go ahead, Shin. Apologies about that.
So if you have the metabolite assessed activity, you know, again, you have to know you have to synthesize the structure. I mean, you have to know the structure to be able to synthesize, so you can really test the activity. So that step still not that automated. Again, we do that. We even just use what you call the bioreactor. That means I use a lot of microsomes and generate enough the metabolite there. Can I assess the extract from there and including that metabolite for activity? But it's just a crude results. Give you some idea. Probably still not that quantitative yet. You know, so depends on project needs. If that is a build project, you probably could do that way. But ultimately, for the IND or special NDA package, you still need to really nail down what is the metabolite, the structure. Then you have to synthesize and test it based on the missed guidance. If your metabolite peak account for about 20% of parent drugs, and you have to measure the activity of the metabolite.
All right, thank you very much, Shin. It seems like the happy hour excitement is already starting. So Abigail is going to give us a few notes about the happy hour. Abigail? So Abigail is muted. Maybe we should unmute her now because she was going to announce a few things about happy hour. Abigail, are you going to announce anything about the happy hour?
Abigail, this is Terry.
Okay, hang on.
Thank you, Shen, for a great presentation and answering all those questions.
Hey, can you hear me? Can you hear me? Oh,
Yes.
I'm sorry. This was a problem that apparently you can't be logged into a WebEx meeting and a Zoom meeting at the same time. So I was getting the happy hour ready, and so sorry about that, everyone. I just wanted to let you all know that the link to the happy hour is in the chat, and every attendee also has the link in their email from me. And I want to thank especially Serene for all of her hard work. Day one was fantastic, and hope to see you guys all over in the Zoom room where we can chat, and then see you again tomorrow. Thank you.
Thank you very much, Abigail, for organizing like a lot of this workshop and the happy hour and so on. So I appreciate your help with that. So I would like to close the session today with thanking all the speakers and the roundtable hosts. I would like to thank you all as well for attending today. I hope you enjoyed the first day, today's program, and found it very beneficial. We have a very exciting program for tomorrow as well. We are looking forward for you all to joining us tomorrow, and hopefully you will enjoy tomorrow as well. Please join us at happy hour, as Abigail says, so that we can relax a bit, network more, and perhaps ask more questions for the speakers. And with this, I would like to adjourn today's program, and thank you all for everything. Bye.
Thank you, Serene.